started. I'll start recording. Thanks everybody for joining for Z Evolution this week. We have a speaker, Geng Xu. Is that correct pronunciation?
Geng Xu.
Oh, Geng Xu, sorry, from University of Nebraska, Lincoln. And it looks like an exciting title. So take it away, please.
Okay. Thanks, Sarah, for the introduction. And hello, everyone. I'm Geng Xu, and I'm a postdoc, work with Dr. Jinliang Yang at the University of Nebraska, Lincoln. Today, I will introduce to you our recent work, Trans-eQTL contributes to gene expression heterosis in maize. So in US, the farmers started planting hybrid corns from 1930s, and about 100% of the cornfield were planted hybrid corns until to 1960s. And with the introduction of hybrids, and as the grain yields was increased dramatically during the last decades. So hybrid corns always exhibit superior performance than their parents. Like here, the F1 shows the silver bigger plant architecture than their parent lines, MO17 and B73. And F1 also have bigger years. So this is this phenomena is called heterosis, also called hybrid vigor. And now there are two measurements for heterosis, middle parent heterosis and beta parent heterosis. For the middle parent heterosis, MPH, people use F1 minus MP and divided by MP. Here the MP calculated by the average value from their two parents. And for the beta parent heterosis, people use F1 minus BP and divided by BP. And the BP is the value from the parents which have the best value. So far, there are three classical models for heterosis. The first one is dominance. And in this model, the heterosis is the result of a complementation of slightly deleterious recessive alleles. And once the F1 have the slightly deleterious recessive alleles combined with their favorable alleles, and they can show heterosis. And the second one is the overdominance model. And here, P1 crossed with P2, and for the first locus, they are the same. But for the second locus, when they combine it together in F1, only one locus, B1 and B2, at heterogosity status can cause heterosis. And the third one is abscesses model. And in this model, non-allelic genes, like here, A2 will interact with B1 and cause heterosis. This is the three models for explaining heterosis. And in previous study, people explored heterosis from many traits, and the heterosis can vary different trait by trait. But most of these traits were collected during the merger stage. So here we want to know in which growth stage the plant can show heterosis. In Kell et al. 2016 paper, they find after 5 days planting, And the hybrids, the B73 and the MOS17, they show a strong performance than their parental lines for biomass and leaf length. So it tells us the heterosis phenomenon can emerge at the very early stage of plant growth stage. So in previous study, people explored the mechanism of heterosis extensively, but most of these traits are agronomic traits they collected during the mature stage, and very few reports on the mechanism of gene expression heterosis. So in this project, we want to address the following questions. We want to know that the gene expression can show heterosis at a population level, and parents with higher gene expression difference, do they can show higher heterosis in the hybrids, and what's the genetic architecture of this gene expression heterosis, and does the expression heterosis eQTL can contribute to the phenotypical heterosis. And to address these questions, our collaborators, Dr. Hongjun Liu, he's from Shandong Agricultural University. He generated the experimental materials. He collected 211 Chinese immigrants, which are from HMAP3, and we divided this 211 immigrants into 20 groups according to the flowering times, and within each group, like here, we generated hybrids using a blocked partial dialyum matting design. And finally, we got about 600 hybrids. And in this project, we collected tons of phenotypes. These phenotypes can be classified as 3 categories. And the first one is the serine trait, and the second is the molecular trait, and the third is the adult trait. So in this slide, I will show you the serine traits we collected. So for the seeds, once we planted in the soil at the first week of planting, and the endosperm can provide nutrition for the plant growth. And the different genotypes, they may have different endosperm. So to exclude the nutrition providing bias caused by endosperm, so in our experiment, we separate the embryo and the endosperm, use a knife, and then only embryo were planted using germination paper to generate our hybrids and their parental lines. And once we have the serine plants, and we collected their fresh weight, dry weight, and mascotal length, seedling length, and the root length, and their total length. And then we also use these young plants and extract mRNA, and we send our samples to the company and generated iron-6 data. And this is our molecular trait. For our iron-6 libraries, we have 204 immigrants and about 600 hybrids. And we also collected the adult trait. We planted the seeds from hybrids and their parental lines in 2021 in 3 locations. And we collected plant height, year height, and kernel row number, year row number, and average grain yield. Because the F1 hybrids always show superior performance than their parents. So once we have the unseq data, we want to know whether the hybrid lines can have more active genes than their parental lines. In this plot, the x-axis is the active gene numbers in each hybrid, and the y-axis is the active gene number in their parental lines. And the different color, the orange color means the active gene number in the maternal lines, and the blue star indicates the active gene number in the paternal lines. And from the picture, you can see most of this dot below the dialogue lines. So it tells us the hybrids have much more active gene number than their parental inbred lines. And once we have different kinds of phenotypes, we calculated middle parent heterosis and beta parent heterosis. And here I will focus on the middle parent heterosis. And for the ciliary traits, you can see here for most traits and most samples, their MPH value is larger than 0. It tells us most of these traits can show heterosis. And we observed similar patterns for the adult trait. And you can see most samples and their middle parent heterosis are larger than 0 for most traits. So next, we calculated middle parent heterosis for our expression data. For each genes, we have their middle parent heterosis data. Here in the plot, the x-axis is the gene ID, and the y-axis, each dot means the media value of each gene expression heterosis. And the red dash horizon dashed line is our threshold. And we found about 60% expressed genes, they have middle parent heterosis greater than 0, this part. Yeah. And we also find about 17 genes with middle parent heterosis greater than 100%. So it tells us a larger number of gene expressions, they have heterosis. Whether this heterosis trait can be heritable, so this is a question. To answer it, we calculated SNP-based heritability. In our formula, we consider the additive variance and dominance variance. And for this ciliary trait, the green bar indicates the SNP-based heritability calculated in hybrids, and the orange bar indicates heritability calculated for MPH, and the blue is for BPH. And for most of this trait, you can see the middle parent heterosis tend to have a higher SNP-based heritability than their trait per se. And adult traits also show similar pattern. and we find a higher middle pattern heterosis for adult treat, a higher SNP-based heritability for the adult treat. And we also calculated the SNP-based heritability for each genes in hybrids, and there are two heterosis traits. For the middle pattern heterosis, the median value of SNP-based heritability is 0.777, and it's significantly higher than that in the hybrids and BPH. It tells us the middle pattern heterosis exhibits a higher SNP-based heritability than their treated state. So next, we want to know how the additive variance and the dominance variance affect this heterosis. Here in this upper panel, I plotted the middle pattern heterosis for each trait, which we collected from different locations, and also including the graph value. And I ordered this volume plot according to the median value of middle pattern heterosis. And the bottom panel is the additive variance and the dominance variance we calculated for each trait in the hybrids. And in this picture, you can see with the increased middle pattern heterosis, and the dominance variance seems increased for the red lines. So we performed the correlation analysis between dominance variance and median value of middle pattern heterosis. We found dominance variance is significantly positively correlated with their heterosis for the adult treat. So next, we also did a similar correlation analysis for our expression heterosis trait. And here we find a similar pattern. The left plot shows the additive variance in the hybrid is significantly negatively correlated with gene expression heterosis, middle pattern heterosis, while for the right side shows the dominance variance is positively correlated with gene expression heterosis. So, and I mentioned at the beginning, there are three models to explain heterosis, and the first one is dominance hypothesis, and it's supposed in the F1 there are deleterious recessive alleles can complement with favorable alleles that can cause heterosis. So we want to ask whether the F1s, if they show higher heterosis, whether they whether their gene can have a higher genetic load for our expression heterosis trait. So here we calculated the genetic load for each gene. Here is the process how we calculate it. For each gene, first step, we extract all these SNPs which are located on this gene, and if for each SNP, if the allele is different from sorghum, we define them as derived allele. Otherwise, it's ancestor allele, and we only consider the derived alleles. In this picture, you can see there are three alleles, A, G, and T. They are derived allele. And then if the SNP with GERP value larger than 0, we define this SNP as deleterious allele. So here, all these we suppose all these SNPs are deleterious alleles, and only the non-ceremonial site were considered. So in this case, we totally have 3 derived deleterious alleles, and we also divided by the gene length. So in this case, the genetic load is 3 divided by 10 kb is 0.3. This is the method how to calculate the genetic load. And once we have the genetic load information of each gene, and we did a correlation between gene genetic load and their gene expression level, and we found the gene genetic load is negatively correlated with the gene expression level. And in addition, we found the genetic load, gene genetic load is positively correlated with the gene expression mirror pattern heterosis. I think this is consistent with the first model for heterosis. Next, we still want to explore which factors can affect gene expression heterosis. Here, we want to know whether higher gene expression difference between 2 parents can have a higher mirror pattern heterosis. Our answer is right. In this plot, the x-axis is the gene expression difference between 2 parents, and the y-axis is the mirror pattern heterosis and beta pattern heterosis. And we find a very strong positive correlation between gene expression difference and their mirror pattern heterosis. So one extreme case of the difference between 2 parent gene expression is single parent expression genes. That means between 2 parents, only 1 parent expressed, and if the gene activated in maternal lines, we call it SPB genes, and if it expressed in paternal lines, we call it SPX genes. In this paper, they found the number of SPB genes, maternal active SP genes, that are positively correlated with the different trait heterosis. Like here, the mirror pattern heterosis for tarsal size and for ear size, they are positively correlated with SPB genes. And we want to check whether we have a similar pattern in our data. So we also counted the maternal active SP genes in our hybrids, and we did a correlation analysis and find the number of SPB genes they are positively correlated with most of the steering trait we collected. Yeah. This result tells us the number of SPB genes that are positively correlated with steering trait mirror pattern heterosis. So GWAS is a very effective way to find genetic loci that control phenotypes. Here we already have different kinds of phenotypes, and we also have genotypes of our population. And then we conduct GWAS for each trait. And here I show one example of our GWAS result for one gene mirror pattern heterosis. And here we find 3 eQTLs. And for the eQTLs we detected in our study, we classified them as Trans-eQTL or cis-eQTL according to the distance between eQTL and their gene. And if one eQTL, the distance between the eQTL and their gene larger than 1 megabase, we call them as trans-eQTL. If less than 1 megabase, we call it as cis-eQTL. Once we have the eQTL information, we find about 75% genes they have cis-eQTLs both in inbred lines or hybrid lines. While for heterosis trait, MPH and BPH, we only find 3.3 or 6.7 genes they have cis-eQTL. This number is pretty small than that in inbred and hybrid lines. And next, we calculate the trans-eQTL ratio for each gene. The ratio I calculated using the number of trans-eQTL divided by total eQTL for each trait. So totally we have about 20,000 genes. So we have 20,000 trans-eQTL ratios for hybrids and to heterosis trait. And this is the trans-eQTL ratio distribution. We find the left two, the distribution is quite different from the right two. The right two is the heterosis trait. For this heterosis trait, we find most of these eQTLs are trans-eQTL. Yeah. So it tells us the trans-eQTL may play a critical role for expression heterosis. So once we have the eQTL information, we find some eQTLs can regulate multiple genes. Here, in this plot, the axis is the eQTL ID, and the y-axis is the regulated gene number for each eQTL, and the blue and the green dots represent the eQTL from MPH and BPH, and we found a lot of eQTLs. They can a lot of eQTLs from MPH and BPH. They can regulate hundreds of hundreds genes, and here the threshold we used is 100, and if one eQTL can regulate more than 100 genes, we call them as a eQTL hotspot. So finally, we found about 900 eQTL hotspot for MPH, and for BP gene expression BPH, we found 290 eQTL hotspots. And we checked this eQTL hotspot across the whole genome. In this plot, we find this eQTL hotspot seems enriched at chromosome arm. So next, we did a correlation analysis between the recombination rate and number of eQTL hotspots for each 1 megabase region. And we found the eQTL hotspots number is positively correlated with the recombination rate. And then we also did a correlation between eQTL hotspot number and nucleotide diversity. And we find that they also are positively correlated. So next, we want to know whether the EQTL hotspots are on the selection during the breeding process. We compiled our EQTL hotspot with the selective sweep, which detected in one et al. Paper. They found hundreds improvement sweeps using only Chinese imbalance. And in this histogram plot, the red vertical lines is our observed value. It means the overlapped number between EQTL hotspot and selective sweep. And the right side is the 1,000 time permutation test, random sample region, compare this region with the selective sweep. And we found the observed value is significantly smaller than by chance. So it tells us this EQTL hotspots were not enriched in the selective sweep, because the EQTL hotspot tend to have a higher genetic diversity. So I think this result makes sense. So here is a summary. And in this study, we found serine root treats, adult treat and molecular treat exhibit a substantial level of heterosis. And we found that dominance variance is positively correlated with heterosis. Maternally active single parent expression genes contribute to the serine trait mirror parent heterosis. And we find that the genetic load of genes is positively correlated with their gene expression heterosis. And last, we found Trans-eQTL plays a critical role in the gene expression heterosis in maize. And I want to thank my advisor, Dr. Jinliang Yang, who gave me a lot of constructive suggestions. And I also like to thank our lab members that gave me a lot of comments for this project. I'd like to thank our collaborators, Dr. Hongjun Liu, Dr. Xuerong Yang, and their lab members. They did all these experiments, and thank them. And I also want to thank Holland Computer Center. They provide us the greater computation resources. I also like to thank the funding support. Thank you for listening, and I will take your questions.
All right, thanks for the talk. I don't see any questions in chat yet, so I'll start off. So I have a question about your maternal single parent expression versus seedling mid-parent heterosis. Yeah, so I'm wondering if you tested for the single parent expression genes, whether the genes are present in both. Like, is it are any of these genes where they're present in one genotype and absent in the other, or are these only genes that are present in both genotypes and only expressed for one parent? I mean.
Here, the SP genes means if one gene expressed in one parent, the FPM should be larger than 1, and in another parent, the FPM should less than 0.1.
Yeah, so what I'm asking is the gene present in both genotypes?
Yeah.
So sometimes like a gene will show presence absence variation.
Yeah. Actually, we didn't have this information like a PAV. Yes. So actually, I'm not sure whether they are PAV for a given gene.
Yep.
Yeah, but it's a good point.
All right, who else has questions? like Jakai unmuted. Were you gonna ask a question, Jakai?
Oh, I have a question about why the hybrids has more active genes.
I think because the hybrids always show superior performance than their parental lines. So this is maybe why the hybrids have more active genes.
Okay, thank you.
Our result is consistent with previous study.
Thank you.
Hi, I'm Shankai at Blockers Lab, and I have a follow-up questions for this. So, why do you think higher expression is better in terms of concerning like active genes or expression heterosis? I can imagine that if in the hybrid some genes got downregulated, that may be good for the higher level traits like the yield or other stuff. So do you have any insights here?
You means you are talking about this slice, high genetic load and higher.
Yeah, if we want to look at the slides, then could you go to the slides for where you show the mid-parent heterosis of the gene expression? And you say that there's 60% of them showing higher expression to the parents. But I'm questioning about those genes that is below 0. Are they contributing to heterosis as well?
I think for a given economic trait, actually, like plant height, it's very complex. It's always controlled by multiple genes. But here, for the unseq data, because some of these genes may contribute to different traits, and I think this result is reasonable. Some gene expression heterosis is below 0.
Yeah, that's my question. So I'm just questioning that. Sorry.
So I'm Jin Liang. That's actually a very good question. So Kremlin et al., when we talk about that small pattern in the Science paper, so the deleterious either reduce the gene expression or cause an abnormal high gene expression level for some of the genes. So those are the pattern we observed in the maize diversity panel, create this small gene expression pattern. So that suggests deleterious alleles not only reduce the gene expression, but also likely create way higher gene expression levels than the regular gene. So that means overexpression not necessarily contribute to heterosis, even if the gene expression is high. For the extreme gene, like the 96 genes that have a mid-parental heterosis way above 100%, those may not contribute to heterosis.
Yes.
On the other hand, so for the left-hand side, those genes with a way lower gene expression as compared to their parents, whether they contribute to heterosis, we don't know, right? Gen didn't test that. That's something we should test for sure. But at least those genes expression pattern is way different from additive.
Yeah. Okay, thank you.
Any other questions? All right, not seeing any. I'm gonna put the video on the Slack, and so if you have more questions, you can reach out and ask Jen more questions later. All right, thank you for presenting, and I will see you all next week.
Okay, thank you.